Before I begin, I'd first like to thank everyone for being here, and secondly, I'd also like to thank the iGEM staff and organizers for preparing this weekend for all of us to come participate in this competition. So thank you. Now, to begin our presentation, I'll introduce myself and my fellow presenter. My name is Terry Vaughn, and this is Brendan Flynn. We are biological engineer majors from Mississippi State University, and our project for this competition is a genetically engineered hydrogen detector. Our current department goal at Mississippi State University in the Biological Engineering Department is to produce oil for the use in biodiesel. When our advisor, Dr. Philip Toe, heard about this competition, he was very excited in the possibility of engineering bacteria to produce biodiesel efficiently. This summer, we quickly formed a team of 10 students and met with this motivation. However, in discovering that this was our first time to participate in this competition, we decided to first form our own team goals and start from there to determine our actual project. Our team goals included learning the procedure to genetically engineer machines, combining the knowledge from several of our departments, which included agricultural and biological engineering, biochemistry, chemical engineering, and electrical and computer engineering. Our third goal was to develop a network between MIT and other iGEM partners for future collaboration. This picture is of our team. Professors I'd like to acknowledge are Dr. Philip Toe of Ag and Bioengineering, Dr. Dinpao Ma of Biochemistry, Dr. Bob Reese from Electrical and Computer Engineering, and Dr. Todd French from Chemical Engineering. This list is of our 10 student members, which include 3 graduate members and 7 undergraduate members. So, keeping our goals in mind for our team, our desired machine function was to create a machine that would detect the presence of hydrogen and would function when turned on by an inducer. The machine would produce a quantifiable fluorescence depending on the hydrogen concentration. Its future uses include the quantifiable detection of hydrogen and could possibly be incorporated in bacterial production of hydrogen in the future. Brendan Flynn will now tell you more about our design and the parts included.
All right. Our composite part consists of 3 previously existing parts from the registry. We used a promoter, a quad part inverter, and a reporter. The promoter we used was listed as a cold shock promoter, but we looked at how it worked and thought that it might also be a hydrogen promoter. We weren't sure, but we found out throughout our testing that it was. The LAC-I, we used the quad part inverter we used was a LAC-I based, and most of you probably know, but a quad part inverter, which is right here, it consists of a ribosome binding site, a gene transcription, a gene site for a repressor, a terminator, and then a promoter that can be repressed by the previously transcribed gene. And then our third part was just a reporter, and we chose YFP. This is our entire constructed part, and I'm going to talk to you a bit about how it works. The uninduced natural state of the LAC promoter transcribes at a very low level, so we get a little bit of fluorescence. Then we used IPTG for our inducer, and it's a very strong promoter when induced, and we get a high level of fluorescence. Now, once we added a low concentration of hydrogen, this turned on the hydrogen promoter, and we transcribed the LAC-I repressor, and the fluorescence began to reduce. The greater in concentration of the hydrogen, the more repressor was transcribed, and the greater the reduction in the fluorescence, as you can see by there. Now Terry's going to talk about testing.
So the testing of our machine required first the introduction of the inducer, IPTG, which provide a broader range of fluorescence levels in the test. It also required us to introduce a variety of hydrogen concentrations and somehow be able to quantify the fluorescence that was produced. We chose to use gassing, which required the cells to be grown in a septum-topped vacuum-ready tube. A vacuum pump was then applied to the tube and used to extract the majority of air from the system. The tube was then filled with the specific concentration of hydrogen that we wanted to test. Photos were taken before application of the hydrogen concentration, immediately after, and 5 hours after the tubes were allowed to incubate. Pictures were taken using a digital camera with standard settings, and the pictures were of the samples that were exposed to light wavelengths close to YFP absorption lengths. This picture depicts the fluorescence of our machine, both unmodified and with the part included. As you can see, the picture on the left depicts the unmodified, while the picture on the right has the part included in it. Our analysis was created using a MATLAB program for pixel analysis. We averaged the pixel value taken in 5 data sets for each sample. The fluorescence change was then calculated for the pre-gassing samples and the samples after 5 hours of incubation, and were zeroed to the unmodified E. coli, otherwise known as wild-type E. coli. This graph shows the results from our test. As you can see, the wild-type E. coli is shown in the blue, and the other shows the comparison to the concentrations of hydrogen going from 25%, 50%, 75%, and 100%. Our results was that we did indeed have a functional machine. It had minimal YFP production or fluorescence without the addition of the IPTG inducer, strong YFP production with the addition of the IPTG inducer, and there was a proportional reduction in fluorescence with the addition of a hydrogen concentration. However, with the successful part, we still have a lot of further testing that we need to endure. This testing includes answering the questions of why the fluorescence in 100% hydrogen was actually higher than fluorescence in 75% concentration hydrogen, and also why positive results could be obtained after 5 hours of testing, but the detection was not immediate, so the incubation was necessary. We'd also like more precise testing with the wavelengths in relation to the YFP absorption. We'd like to give acknowledgments to our advisors, the Mississippi State Bagley College of Engineering, the Mississippi State College of Agriculture and Life Sciences, as well as the iGEM ambassador, James Brown, and the rest of the iGEM staff. Our future ideas for entry into this competition include controlled lipid synthesis to produce efficient energy sources from inefficient organic sources, water splitting for efficient hydrogen production, tar digestion to yield cleaner energy sources, and insulin or blood sugar regulations for diabetes control.
Before we go into questions, I'd like to clarify one thing. For our results, the blue bar is the 0% hydrogen concentration, and the 100% was the one that we weren't quite sure about why there was more fluorescence there, but since it was just preliminary testing, it could just be something as simple as the error involved. That would require much more testing to fully confirm all the way from 0 to 100% that our promoter works consistently. And now we're going to go into questions. We're going to ask Victor Ho and Robert Morris to come up and help us with the questions. Okay, the question was, is oxygen a factor in the changing in fluorescence? And while it's possible, that was one range of tests we haven't run yet. We'd like to run tests where we change the other composition. You know, if we have 50% hydrogen, what do we use? What else do we use in there? We use air. We used CO2 once, and we got poor results. And so we've used air because that was a combination that gave us good results once. But I'd like to try, as you said, with a few different, maybe nitrogen and... Yeah. Okay. Back there? Okay. The question is a more detailed description of the hydrogen promoter. I'm going to ask Victor.
Okay. We just cloned this promoter from a hydrogenase. The other people doing this this work, and the test, the hydrogenase will be turned on by the hydrogen. Then so that's what is our idea come from. So we just think about maybe this promoter will be turned on when you add the hydrogen to the cells. So just clone this promoter. Then the detailed biochemical reaction, we don't have really many idea about that one.
And the question is, are we sure about the relation between intracellular and extracellular hydrogen concentrations? Hydrogen perfuses very well through most materials, so we were assuming a pretty consistent level, but anything to add on that one? We don't know for sure yet. I'm sorry. Right. Currently, not very. They have mechanical hydrogen detectors that are they don't require the waiting period, and you can get digital readouts. So as of now, the use of ours specifically isn't competitive, but possibilities in the future, maybe. The question was, what kind of environments would we be testing the hydrogen in? And our department has a large focus on biofuels, and so we were looking to implement it in different forms of bioreactors, or something. Maybe as one of our future goals is producing a nano machine that would produce, using bacteria to produce a biofuel. Maybe we could incorporate the hydrogen detection into that bacteria. So it'd be very, you have your box bacteria, and you can just tell when it's working. Yes. Mm-hmm. Okay, the question was the time frame when we took the pictures for our testing. We ran 2 or 3 tests, and as I said once, we used the CO2 instead of the air. But when we did use the air, we ran 2. One time we took pictures much later, and we had no real results. At that point, the cells had died. They don't last very long, and the ones we're using at least. But 5 hours seemed to be a good amount of time for the hydrogen to work its way in and the YFP production to fully implement. Yes. The question was using alternative quad-part inverters, maybe, or ribosome binding sites to not require the IPTG. I think in our initial design, we wanted to have the IPTG to kind of have a control over our part, so we could have a with IPTG or without, just for an extra comparison in our testing. But it could definitely, now that we think that we've shown preliminarily that the promoter also promotes for hydrogen, any number of other designs could be used.
Yeah, you mentioned that you use with IPTG, but it wasn't really I imagine it's because it activates the lactate. I was wondering, is there like a specific promoter, or how does that work?
You're asking how the IPTG induces?
Yeah.
Okay, well, okay. IPTG has 2 functions. One is to actually remove repressors, and the other is to induce. I don't know the specifics on the small scale of the induction, but I know.
Is it possible? Because I know normally it inactivates black IPT. So is it possibly some basal transcription that from the other machine that's inactivating and thereby
The question is there another gene affecting the basal levels along with the IPTG? And we're not sure. Good question. Okay, thank you. Everybody needs to go outside for the picture now.